Okay, so we're going to start with review. As I mentioned, I'm not going to answer in enormous details to every question. Obviously, we won't have time for that. Some of those questions are simpler, some of them harder. I'll give an idea. So functionally, obviously, know that it's a regulatory system, right? Structures, I told you the glands that you must know from like up head to toe. We're talking hypothalamus, pituitary, and pineal glands up here. Then we talk about thyroid and thymus. Then, come on in. Thyroid and thymus. And then we talk parathyroid, in the back of thyroid gland, pancreas, adrenal, gonads. Did I miss any of the primary? I don't think so. I think we're good. Understand hormones are endocrine because they're released in the blood. That's pretty much the difference between these two. Are we good? Now, as I go, for two things. First, tell me if I go too fast. Second, if you have a question, like question, just interrupt me. Don't raise your hand, just holler. Okay? Primary endocrine organs use hormones, obviously. Know which ones are primary. Now, when I tell you know which one are secondary, if I give you a list, you have to recognize which one is primary, which one is secondary. Same as we did on the quiz. So I kind of asked the same thing multiple times. The main difference is that secondary has some other function. Well, it's nervous and endocrine. Be able to compare. Okay? Nervous is shorter lived effects, right? But effects are faster. For nervous system, the target cells will be neurons, muscle cells, and sometimes gland cells. For endocrine cell, it's practically every cell in the human body, wherever blood reaches. Chemical mediators, neurotransmitters for nervous, hormones for endocrine. Site of action, again, remember, nervous system is more local. It's either synapses. Synapses with neuron synapse or neuron muscle cell synapse. Usually it works like this. Okay. Two major types of hormones. I'm referring to fat versus water soluble. For water soluble, remember we have amines, peptides, proteins. For fat soluble, we have sterol hormones, like cholesterol-based hormones, pretty much. We have, thyroid hormone, and we have eicosanoids. Those three, okay? So, lipid, we have sterols, right? We have thyroid hormone, and we have eicosanoids. So, those are three types, okay? Yes, I can ask you, which of the following hormones is water soluble? Or which of them is lipid soluble? Yes. You have to understand the major difference, which is ability to cross the membrane. Lipid soluble can, water soluble cannot. So, the importance is that water soluble are going to have receptor on the surface, right? Versus lipid soluble hormones that are going to have receptor in the cell. Does that make sense? Now, look, what I do, I quickly structure what we've already learned. Okay? So we go deeper and deeper in the details. We go from, big picture to small details. So now, for water solubles, I ask for three major mechanisms. Okay? They interact with a surface receptor Okay? And then we have here, I refer specifically to G protein coupled receptor. G protein coupled receptor. Except for hormonal axes like HPA, HPG, well, I will tell HPA, HPG, HPT. If you have an acronym, ask me. If you're not sure about an acronym, ask me. I will tell you what it means during the exam. So, 3 mechanisms. One is for cyclic AMP. So, I have binding to receptor, activation of G protein, activation of adenylate cyclase, synthesis of cyclic AMP. And cyclic AMP will activate proteins in the cell. That's all you need to know. Am I clear? Second will be binding to the receptor, activation of G protein, activation of phospholipase C, synthesis of IP3, release of calcium from endoplasmic reticulum. That's it. And third mechanism is tyrosine kinase-based receptors. Hormone binds the receptor, activates tyrosine kinase that is the part of the receptor and is in the it's in the cytoplasm anyway. So tyrosine kinase phosphorylates the proteins, and you have like the whole thing going on in the cell. Please know what kinases do. They phosphorylate. That's all you got to know. That's it. You phosphorylate. Okay. Lipid-soluble hormones. Highlights. Receptors inside of the cell. Okay. So if I tell you, can it be in the nucleus? Yes, in the cytoplasm. Of course. It's number one. Number two, once there is a complex, hormone, remember, hormone binds to the receptor, not to the protein, not to the DNA, not to the RNA, to the hormone binds to the receptor protein. Does that make sense? When hormone makes the complex with the receptor, this complex binds to DNA in the nucleus, and then you have DNA is transcribed into RNA, RNA is translated into the proteins. Does that make sense? That's all you got to know. Three stimuli for the hormone production: humoral, nervous, hormonal. When I say mechanism, I'm not, don't overthink it. Did you remember the question on the quiz where you had to match the stimulus type with a description? You're going to have that, well, question like that on the exam. So you have a description of the stimulus, and you have to match the type of stimulus. That makes sense? So like for ADH, osmolality changes will be humoral. For growth hormone, stimulation by gonadotropin growth hormone releasing hormone will be hormonal. Okay? For corticotropin releasing hormone in the hypothalamus, stimulation will be nervous. Does that make sense? I explain you what's going on, you match. Any questions on that? Not really? Moving on. This, 3 ways of hormonal action. Autocrine, hormone acts on the same cell. Paracrine, hormone acts on the adjacent, closely located cell. And endocrine, out there. The hormone is transported with the blood to remote location in the body. We cool? Hormonal regulation occurs through negative feedback, except for the one that we really talked about is oxytocin. Know about oxytocin. Am I clear? This one, yeah, know that it's oxytocin is regulated by positive feedback. When I say what is negative and positive feedback, by the way, I'm not going to ask you what is negative or positive feedback in the exam, but you have to be able to recognize one if it pops in the question. So if you come to me and say, is that an example of positive feedback or is that an example of negative feedback, I will not help you. Am I clear on that? So you have to know, you have to be able to see. Positive feedback amplifies the initial changes. Negative feedback decreases initial changes, brings the system back to the homeostasis. Am I clear? Radioimmunoassay. Non-radioactive hormone from your sample substitutes radioactive hormones in the complex with antibody. And then you measure radioactivity of either supernatant, the fluid that contains hormones that were substituted, or radioactivity of the complex of antibody with a hormone that you can pull down. For this pretty much allows you to work your way through the method as the concentration of non-radioactive hormone goes up, okay? The concentration of this complex, okay, between your experimental hormone and the antibody, it goes up as well, okay? Complex between antibody and radioactively labeled hormone, the opposite goes down. That example with the fans of two teams. When you increase the number of, like, I don't know, Manchester City fans, the number of Liverpool fans in the stadium that can see it, can be there, will go down because there are only so many seats. Precipitation, what's the purpose of the precipitation? To measure how much complex we have between the antibody and hormone, so we can tell, okay, we have so much radioactivity, which means so much radioactivity was lost, so we have so much non-radioactive experimental hormone. Supernatant is what's above the precipitate. What's left in supernatant? Unbound radioactive hormone. How does the radioactivity of the supernatant changes with the increase in concentration of non-radioactive hormone? The more non-radioactive hormone you add, the more hormone you have in the sample, the more unbound radioactive hormone will remain in the supernatant above the antibodies. Okay? For ELISA, what you got to know, plate has antibodies. You had hormone. Hormone binds to those antibodies. And then you had the secondary antibody, which will detect the hormone. Secondary antibody. What is common for these two assays? Both cases, both assays, the main idea is binding of a hormone with a specific antibody. That's why it is so specific. Okay. So that's going to be primary antibody, that's going to be hormone, it's going to be secondary antibody. Essentially, if you have a plate, that's primary antibody, that's the hormone, that's the secondary antibody. Okay. hypothalamus. As I mentioned, you don't have to list anything. It's going to be multiple choice. So, what does it produce? Releasing and inhibiting hormones. When we start. Say again? So, thyrotropin releasing hormone, corticotropin releasing hormone, growth hormone releasing hormone, growth hormone inhibiting hormone, gonadotropin releasing and gonadotropin inhibiting ones, prolactin inhibiting factor. That's pretty much it. And look, this is really easy. If the name contains releasing or inhibiting, it's hypothalamic. Warning you, I may make up a hormone that doesn't really exist. Okay? So, it shouldn't be the only criteria, but, you know, if you see corticotropin inhibiting hormone, no. No. This one just doesn't exist. Does that make sense? Now, let me not, please, let me not focus on the stimuli now. Because when we're going to talk about each individual axis, we'll have plenty of stimuli. Does it make sense? Do you understand what I'm trying to say? So obviously, just brief example, obviously, we talk about cortisol. What is the stimulus for cortisol? Stress. So what is the stimulus for corticotropin releasing hormone all the way up in hypothalamus? Stress. So we kind of can merge it. Does that make sense? So we're gonna get there. For prolactin inhibitor, it inhibits prolactin. Okay. Stimuli and functions for all other hypothalamic hormones we will mention down the road. Good? Any questions? We can move on. Where's pituitary gland? The purpose of this question is so you can recognize it on the picture. Inferior to hypothalamus, connected by infundibulum, lies in the sella turcica. Infundibulum, sella turcica, which two parts? Anterior and posterior, adenohypophysis and neurohypophysis. Which another endocrine gland controls pituitary gland? Hypothalamus. Good? So far, we're clear. The route for hypothalamic hormones to get to the anterior pituitary. Primary capillary plexus, right? Neuroendocrine cells in the hypothalamus release hormones in the primary plexus. Then hormones go through the portal system and end up in the secondary plexus. From the secondary plexus, they diffuse into the anterior pituitary and stimulate whatever they're supposed to stimulate. Are we clear? Know the name of that system. Hypothalamo-hypophyseal portal system. If you see portal, it is vessels. Always. Any portal system consists of blood vessels. Hormones of the anterior pituitary are released into the circulation in the anterior pituitary. That's it, in systemic circulation. Hormones that are produced by anterior pituitary gland. Let's try it. Growth hormone. Thyroid stimulating with thyrotropin. ACTH or corticotropin. Follicle stimulating and luteinizing, which are gonadotropins. There's melanocyte stimulating hormone, but we didn't talk about it, so it's not going to be on the exam. I think we're done. I think that's it. For anterior pituitary, okay? Again, there will be no list, so don't worry about forgetting anything. Okay? I mean, remember them, but you don't have to list them all. Okay, growth hormone. Let's stick to it. First, what is the axis? What does it start with? Okay, stimulus. We can have low blood sugar, we can have low free fatty acids, or high amino acids, or trauma, exercise, sleep. Think about this, okay? So, pretty much. Okay, we'll get to the effects. Okay, now, in terms of regulation, what is the first hormone in the axis that's produced? GHRH, growth hormone releasing hormone. It's produced in the hypothalamus, acts on anterior pituitary, which releases growth hormone. What are the direct effects of growth hormone? Direct effects of growth hormone. Elevate. So, if blood glucose is low as a stimulus, what's going to be the direct effect? Increase the blood glucose. If free fatty acids are low, what's going to be the direct effect? Increase free fatty acids by breaking down fats, so lipolysis. What are indirect effects? Protein synthesis, cell proliferation and growth, skeletal and muscle growth. Okay? So, protein synthesis. It will use those elevated blood amino acids. Does that make sense? Got it? High blood amino acids, protein synthesis, blood amino acids will be taken into the cell and used for protein synthesis. Why those effects are called indirect? What are they mediated by? Liver. What kind of another hormone does liver produce? IGF. Insulin-like growth factors. So essentially you have this, then this, I'm sorry, and then this, right? Does that make sense? What are the effect of IGF on these two? Exactly. So this is your negative feedback. Does that make sense? Now, I'm not going to do the run through for every axis, but please, if you can picture this, then you can answer questions like, if we increase the concentration of growth hormone, what's going to happen to the concentration of IGF? It's going to go up. Does that make sense? If we increase IGF, like we start shooting IGF, what's going to happen to growth hormone or GHRH? They're going to go down. Does that make sense? There will be questions like that. Please know them. I may combine more than one effect in one question. So you have to break them apart, each answer. I may ask you about effects on the growth, or effects on metabolism and effects on hormones. Do you understand what I'm saying? So if I tell you, in the person who injects growth hormones as the performance enhancement drug, what do you expect to see in terms of blood sugar? It's going to go up. Blood fatty acids. It's going to go up. IGF. They're going to go up. Does that make sense? Recognize those things. I focus on metabolic effects on the hormones. Where there are any significant ones that we mentioned extensively, as they are. So, stimuli, as we mentioned, low blood sugar, low blood free fatty acids, high amino acids, stress, trauma, exercise, sleep. That's pretty much it, what I want you to know. On what will inhibit? High sugar, high blood free fatty acid, low blood amino acids, obesity, and aging. Does that make sense? We talked about those things. Any questions before we're moving on? Let me see that. Okay, we covered this. Covered this. We covered. Okay. Remember that in direct effects, yeah, we got this. We got changes in blood glucose. We got this. So increased blood amino acids will increase. Increase, decrease. Testosterone and estrogen will increase. This one will decrease. Okay, but I'm going to be candid. Testosterone, estrogen, not yet. I'm not asking about this. Okay, don't worry about testosterone, estrogen. IGFs, yes, absolutely. You know that you have to know that IGFs inhibit growth hormone. Did I make myself clear enough? Okay. But abnormalities in the childhood to low dwarfism to high gigantism. In the adulthood to low lethargy, there's no really like a specific term, to high acromegaly. Okay? How can it be used in sports? Performance enhancement drug. Consequences? Acromegaly, overgrowth of stuff. Diabetes, type 2 diabetes because of elevated blood sugar, you know. Thyroid. Again, when I tell you where it's located, these fellas are going to be there. It's kind of a cheat right there. You can see thyroid gland in those. Okay. What I want you to understand, you have two lobes connected by isthmus, the neck thing, sort of. So the inferior to the larynx, anterior to the trachea, elementary building blocks, follicles filled with protein-rich colloid, two types of cells, follicular and parafollicular, cuboidal epithelium. Follicular produce thyroid hormone, parafollicular produce calcitonin. It's kind of same thing going over and over again. Okay? Does that make sense? Well, three hormones, T3, T4, it's actually two types of thyroid hormone, and calcitonin. Calcitonin is water soluble, T3, T4 are lipid soluble, tyrosine is required for thyroid hormone, iodine too. So far pretty straightforward, isn't it? Now, iodine, where is it first? Where does it come from? In the body. Huh? Okay, from the outside it's a component of salt. Now, in the body, where does it go to first? The B word. Blood. From the blood, it gets into the cells, follicular cells. In the from the follicular cells, iodine enters the follicle where it binds to the tyrosine. And after the series of tyrosine, is the amino acid tyrosine, is it separate or is it a part of a protein? Part of a protein. So when it binds to the tyrosine, eventually tyrosine becomes thyroid hormone. Is it separate or still a part of a protein? Yet. Still a part of a protein. Then from the follicle, and the protein is thyroglobulin. So that thyroglobulin gets into the follicular cell. And what happens to thyroglobulin in the follicular cell? it is broken down, so thyroid hormone is released into the bloodstream and everybody's happy. Does that make sense? Understand? What I told you are the major steps that you need to know. The biosynthetic pathway, screw that. Okay? But like tyrosine, iodine, and in the end you have thyroid hormone. That is yet attached to the protein and will be released only after the breakdown in the follicular cell. Are we clear? So pretty much we can kind of just know the sorry. So like the final thing, you know. No final product, which is T3, T4. Thyroid hormone is released into the blood. Stimuli. What stimulates production of thyroid hormone? Low BMR. And what does it do to BMR? It increases it. Again, you have axis, which consists of TRH in hypothalamus, TSH in pituitary, and thyroid hormone in thyroid gland. Does that make sense? What effect thyroid hormone will have on TSH and TRH? Huh? It will inhibit them. Does that make sense? So, say somebody has okay. Let's talk about pathology because it's much easier, really, to appreciate the role. So, first of all, low iodine. What's going to happen to levels of thyroid hormone? Low iodine. I just can't hear you, so say louder. No iodine in the diet. What's going to happen to the levels of thyroid hormone? Decrease, because we can't make it. We need iodine, there's no iodine, there's no thyroid hormone. What's going to happen to TRH, TSH levels? Huh? They're going to go up, both of them. There you go, that's your clinical picture. What's going to happen to thyroid gland? Enlarge. Why? Huh? Of what? Nope, there is no thyroid hormone. The one above, TSH. It will stimulate thyroid, kind of, you know, let's grow and make more. Well, it will grow, but it won't be able to make more. It's compensatory effect. Does that make sense? Okay, so that's endemic goiter or nutritional goiter, right? Hyperthyroidism. The most common cause is Graves' disease. Autoimmune. So you have special antibodies that stimulate what? There should be another word after TSH. They stimulate TSH receptors on thyroid gland. Does that make sense? So they mimic TSH. Do you see what I'm saying? Those antibodies are like TSH, so they stimulate thyroid gland to make thyroid hormone. So in hyperthyroidism, levels of thyroid hormone are high. Levels of TSH and TRH are low because of the inhibition. What are the physiological symptoms? Huh? Hyperactivity, heat, intolerance. Cardiovascular symptoms, increased heart rate, huh? Increased blood pressure, yes. Generally nervousness. Usually people are lean, so it used to be used as the weight loss treatment. It's still, as far as I know, it's still used, but it's not really a brilliant idea to do that because you mess up the entire endocrine regulation. Does that make sense? Okay. Now in Hashimoto, which is hypothyroidism, what's going to be the clinical picture? you can say just opposite. That's it. So, thyroid hormones low, TSH, TRH high, obesity, lethargy, cold intolerance, low blood pressure, and everything just flipped. Does that make sense? I will ask you about the regulation of the axis, the effects, and in terms of the stimulus, BMR is fine. That's it. Effects, know these basic effects: cardiovascular, heat, cold, tolerance, intolerance. Okay, that's what, and metabolic effects, pretty much obesity in hypothyroidism versus weight loss in hyper. Did I make myself clear? Good. And I'm telling you, here, if you can draw the axis, then it's going to be much easier. And of course, you can draw on the exam. Oh, yeah. Thyroid hormone, lipid soluble or water soluble? Lipid. So where's the receptor? In the cell. So the complex between the receptor and the hormone will bind to, that's all you need to know. Well, DNA, and then you have RNA, and then you have protein eventually. You don't need to know like retinoid X and all that kind of stuff. So it's kind of detailed. Effects of thyroid hormone. Will they be generally, like those genomic effects, are they going to be fast or slow? If it's, slow, because in the cell. And then you have to bind to DNA, and then there should be genes transcribed, and then proteins, synthesized. It's just the whole ordeal. It takes really long. Does that make sense? Cross-dietary goiter. How can it be treated? Cured? Iodine. Hyperthyroidism. Cause of it. Autoimmune. Those antibodies. That's what we just talked about. Effects on the HPT axis hormones. Thyroid gland increase. Why does thyroid gland increase in Graves' disease? Constant stimulation just keeps growing because of those antibodies that mimic TSH. Hypothyroidism, talked about it. Myxedema, lack of thyroid, which leads to, all you need to know, mechanism. Accumulation of water, well, in the form of a gel in the subcutaneous tissue. All you need to know. Am I clear? That's all you need to know. Questions, concerns? No? Good? Call 1-800. I don't care. Parathyroid gland. This is simple. So, location-wise, back of the thyroid gland. Two types of cells. Chief cells, or parathyroid cells, and oxyphil cells. Chief cells produce parathyroid hormone. Low calcium, parathyroid hormone goes up. Blood calcium goes back to normal. Blood calcium elevates. Am I clear? Okay, we're good? Three mechanisms of getting calcium in the blood. One, from the bones, stimulation of osteoclasts. Two, in the kidneys, increased calcium retention in the kidneys, so less calcium is excreted. And third, increased absorption of calcium in the intestine. What do you need? Is that a direct effect of parathyroid hormone or not? It is not. What do you need for it? What? Vitamin D. Vitamin D is activated in the kidneys, right? Acts on the intestines, calcium absorption increases. Are we clear? That's all. That's all. Calcitonin, parafollicular cells of thyroid gland. High calcium in the blood, increased calcitonin, leads to the deposition of calcium in the bone. Calcium levels in the blood tightly, loosely regulated. Tightly. Okay. Now, hypocalcemia, contractures of the muscles. Hypercalcemia, well, mostly muscle problem. Hypercalcemia, renal, kidney stones, and muscle weakness. Adrenal glands are on top of the kidneys, 2 layers, medulla and cortex. So medulla produces water-soluble epinephrine, norepinephrine, right? Cortex produces lipid-soluble, different lipid-soluble steroid hormones. 3 layers, glomerulosa, fasciculata, reticularis, okay, from top to bottom. Now let's talk about, so glomerulosa produces aldosterone. What are the functions of aldosterone? 2 main stimuli. Low BP and high potassium. Okay. So if blood pressure is low, what does aldosterone do? Increases water retention in the kidneys, right? Increases blood volume. It's actually via renin. So you have renin stimulates angiotensin, angiotensin stimulates aldosterone. And aldosterone in the end, increases sodium retention in the kidneys. Then water follows sodium, increased volume of the blood, increased blood pressure. Does that make sense? Okay. Again, I'm not asking you to describe it, but be able to recognize the right sequence of events. Okay. Potassium, if aldosterone goes up, potassium is excreted, right? And if aldosterone goes down, then potassium is spared, contained, sodium is excreted. I said name 3. Oh, the third. Potassium, low blood pressure, stress. General psychological stress increases aldosterone production, which leads to question? Mary? High potassium. So it gets rid of it. Increased aldosterone production, high blood pressure. So blood volume goes up, blood pressure goes up, potassium concentration goes down. This is renin and angiotensin. Renal consequences of elevated blood pressure. Oh, that's so complicated. Okay. If blood pressure goes up, what happens to aldosterone levels? It goes down. What happens to the sodium retention in kidneys? Sodium is excreted. When sodium is excreted instead of being retained, what is excreted with sodium? Water, natriuresis, right? So you produce urine, which contains a lot of urine, which contains a lot of sodium. Does that make sense? What happens to the blood volume? It goes down, blood pressure goes down. Stimulus for atrial natriuretic peptide. High blood pressure. Atrial natriuretic peptide inhibits aldosterone. And there you go. So the subsequent physiological effect is natriuresis and lowering of blood pressure. Aldosterone is fat soluble. Receptors in the cell. Hormone goes in, the complex goes into the nucleus, binds to DNA, and expresses the genes that help to retain sodium. So essentially, the pump, sodium-potassium pump and sodium channels. Does that make sense? Excessive aldosterone on the blood pressure, potassium levels. If I ask you if somebody has intrinsically high levels of aldosterone, you tell me potassium is going to be high levels of aldosterone. Potassium is going to be in the blood low. Blood pressure is going to be high. Blood volume is going to be high. If potassium is low, it's going to lead to muscle weakness. Potassium levels are high. If aldosterone is very, very low, then potassium levels in the blood are going to skyrocket and the person will probably die because of the ventricular fibrillation. Does that make sense? Cortisol, the major stimulus. Stress. Hormones, CRH in hypothalamus, ACTH in pituitary, cortisol in a zona fasciculata. How is negative feedback established in HPA axis? Well, elevated cortisol levels, how they affect levels of ACTH and CRH. They decrease those levels. Are we cool about it? Okay. That's your negative feedback. I will be focusing on metabolic effects of cortisol and immunological effects of cortisol. What does cortisol do to the immune system? Suppresses it. We get it out of the way. Metabolic effects. The main metabolic effect of cortisol is elevated blood sugar. Okay? So, will it stimulate glycolysis in the cells? Breakdown of glucose in the cells. Will it stimulate breakdown of glucose in the cells? If it elevates blood sugar, will it stimulate glycolysis? No, but it will stimulate glycogenolysis and gluconeogenesis. Does that make sense? Or lipolysis to break down fat and eventually convert it into the glucose. Does that make sense? What will it do to proteins? Come on, 50-50 question in jeopardy. Can I either make them or break them down? Break them down. Protein catabolism will be increased because amino acids can be a valuable source for glucose synthesis. Does that make sense? No metabolic effects of cortisol. Please, 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 please. Okay? That make sense? So in chronic stress, when blood glucose goes up all the time, what's going to happen with those patients? Obesity. Well, I would bet on type 2 diabetes first, but they will gain weight absolutely. Yeah. Why is stress often accompanied by elevated blood pressure? Stress stimulates what? Aldosterone. Okay. Because it all acts on the adrenal gland. Receptor for cortisol is in the cell. It's kind of same stuff. I'm not going to ask you about what is the name of the receptor. Am I clear? But I can ask you, what is true about cortisol? And you have to remember it's a fat-soluble hormone, so a receptor will be inside of the cell. Am I clear? Jesus, it's immunosuppressed. Results from overexposure. So high cortisol leads to disease, like the name of syndrome, Cushing's, which is characterized by obesity and specific fat accumulation, buffalo hump in the moon phase. The deficit of glucocorticoids is Addison's. Now, if I tell you have a patient with Cushing's disease because of, I don't know, adrenal tumor, okay? Cortical tumor in the adrenal gland. So the patient constantly overproduces cortisol. Clinically, in the blood, what you're going to see? CRH will be, if cortisol is high, CRH is going to be, come on. So CRH is going to be low because cortisol will inhibit it. Does that make sense? And ACTH will be low as well. Does that make sense? You understand what I'm saying? Blood glucose in those patients will be high. Good? Understand those things. Reticularis androgens, their main function in men is not known. In females, substitution for decreasing estrogen after the menopause. Adrenogenital syndrome, please. I just love this regulatory moment, so please know this. So what does adrenogenital syndrome start with? The patients don't have cortisol at all. What happens to the levels of CRH? They go up. ACTH goes up. ACTH stimulates adrenal gland, increases levels of androgens produced by the zona reticularis, ambiguous sexual characteristics. Does that make sense? Masculinization in females and the word just completely escaped my mind, so I apologize for it. Just don't remember, just speechless. But bisexual genitalia in males. Okay, I see this word, but I can't read this. Okay, just escaped. Okay. Hypospadias in male. Posterior pituitary gland, does it produce hormones? No, it stores two hypothalamic hormones, which are oxytocin and ADH. Oxytocin should be pretty straightforward. Oxytocin, uterine contractions, stimulus back to the hypothalamus, more oxytocin, hypophysis releases more oxytocin, more contractions, more oxytocin, just goes back and forth. Are we clear on that? Just don't want to spend too much time on that. How the hormones from hypothalamus are delivered to posterior pituitary via hypothalamus hypophyseal tract, which, say again? Tract, which is pretty much axons of the nerve cells. Does that make sense? Oxytocin, role oxytocin in men, supposedly, maybe, bonding, maybe not. Antidiuretic hormone. Okay. What are triggers for antidiuretic hormone? Low blood pressure. What else? High blood volume will be high blood pressure, right? So low blood pressure means the volume is actually, low blood pressure means that the volume is actually low, which means the concentration of solutes in the blood is high. The volume is low, but the concentration is high. So low blood pressure, low blood volume, high osmolality, which may result from dehydration. So what does antidiuretic hormone do? Entire chain of physiological effects in the kidneys. Come on, give me a break. So what does it do? Antidiuretic. Retains, no, not the sodium. It retains water directly. How does it retain water? Not altogether. And speak up. Osmolality actually remains pretty much the same in the kidneys. It increases the expression of certain proteins. Channels, remember? Proteins called aquaporins have more. If somebody said it, I appreciate it. I just didn't hear because you were too quiet. Aquaporins in the kidneys, the more aquaporins you have in the collecting ducts, the more water will leave the urine and come back into the kidneys and, end up in the circulation. Does that make sense? So, high osmolality, more aquaporins, more water retention. Your ECF, your blood becomes diluted by that incoming water. Osmolality goes back to normal. ADH is not produced anymore. Does that make sense? So if we jack up ADH production, what's going to happen to the urine volume? We jack up. We increase ADH. Increase antidiuretic hormone. The volume is going to decrease. What about the concentration of the urine? Concentration of the urine is going to go up because you take all the water out. Does that make sense? If you inhibit ADH, exactly opposite. Does that make sense? Okay. So pretty much ethanol that inhibits ADH leads to dehydration during the excessive drinking if you do not compensate. From personal experience, before go to bed, compensate. Like 3-4 glasses of water before you go to bed really help the next morning. Diabetes insipidus. Where does it come from? Underproduction of ADH. So people don't retain water, they're constantly thirsty, they lose enormous amounts of water. And it's pretty much just flushes through them. They don't lose really any solutes because of that. Okay. Pancreas. I'll show you the picture. Recognize the pancreas. Okay. Yes. Sure. Uh-huh.
When it produces, does the water retain in the kidneys or in the stomach?
Okay. In the kidneys, before urine goes in from the kidneys to the ureter, pretty much the last step for the filtrate in the kidneys to become urine is to go through the collecting ducts, which are tiny little tubes. Let me finish and then you will tell me if I answered your question or not. Okay? Listen. The walls of these collecting ducts consist of epithelial cells. When ADH increases, those epithelial cells get way more aquaporins that will channel water from the filtrate through the cells into the kidneys and eventually into the blood. So, water retention occurs in the kidneys. When aquaporins are increased in the kidneys retain more water. Did I answer your question? I think you're overthinking it a lot.
Yeah.
Focus on the organs. We will talk about more specific cellular effects of ADH when we will talk about renal system. So far, know that those are water channels that help to retain water in the kidneys. Did I answer your question? Good. Pancreas, 3 anatomical parts of the pancreas. Well, head, body and the tail. Tail is here on the side. Just remember, it's closer to the spleen. That's where the tail is. That is on the opposite side. So, structures in the pancreas that produce hormones, Langerhans cells. You may see them on the exam as a slide or as a picture. Am I clear? Just recognize them. That's one of your anatomy questions, maybe. Okay. 4 types of cells. Alpha, beta, delta, PP cells. So, what you need to know, alpha cells produce glucagon in response to low blood sugar. Physiological effect, elevated blood sugar. Beta cells, insulin in response to high blood sugar, effect, low blood sugar. Delta cells, somatomedins, regulation of insulin production from the kidneys. Okay, stimulus, starvation. PP cells, I'm not going to ask you about this. Just don't worry about it. Okay, insulin is a protein. So the receptor is where? Surface. Receptor, now, sequence of events that triggers insulin production. First of all, what is the initial stimulus? High blood sugar. So glucose enters beta cells. What is it used for in beta cells? In which cellular process? Glycolysis first. ATP is produced. ATP closes potassium channels. Potassium stays in the cells and leads to depolarization of beta cells. Depolarization opens calcium channels. Calcium flows into the beta cells and insulin is released. Now, insulin production metabolically leads to, that's just like the whole thing. So, what is increased as the result of insulin production? First, glucose, uptake of glucose by the cells. And when glucose is in the cells, what's going to increase in the cell? Which biochemical processes? Glycolysis, glycogenesis, and lipogenesis. We're good? Anything that will utilize glucose. Does that make sense? So for glucagon, it will be exactly the opposite. Instead of uptake, glucose will be released from the cells, right? And the processes that will be elevated will be go ahead. Glycogenolysis, breakdown of glycogen, and gluconeogenesis, right? So you have to think, in case of high blood glucose, you want to use it. Any process that can use it will be upregulated. In case of low blood glucose, you want to make it. Every process that makes it will be upregulated. Does that make sense? Glucagon is a protein, so the receptor is on the surface. Okay, cellular mechanism of insulin action. All you need to know, again, the processes that it stimulates. Am I clear? Am I clear? So you need to know that uptake goes up, ATP goes up. That pretty much refers to the mechanism of mechanism of the insulin release from the beta cells. So once beta cell is depolarized, calcium channels are open, calcium goes into the cell, release of insulin from the cell, and obviously the more glucose you have in the blood, the more glucose gets in the cell, the more ATP is produced, the faster potassium channels will close, the faster the cell will get depolarized, the more calcium will get in, the more insulin will be released. That makes sense? So that's your dose-dependent relationship between insulin and glucose, because the more glucose you get, the more insulin you're going to produce. Insulin interacts with the receptor, which is on the surface. Tyrosine kinase is activated. Any pathways that will lead to glycolysis, lipogenesis, glycogenesis, and glucose uptake. Does that make sense? That's what we just talked about. Can muscle cells uptake glucose without insulin? No. What happens to the cell metabolism in people with diabetes? People who are insulin deficient, they switch to metabolizing which source? Instead of carbohydrates? Fat, because they can't take carbohydrates in. And that leads to ketosis due to the accumulation of ketones. Physiological effects: blood pH goes down, blood becomes acidic. Consequences: eventually coma because of the depression of neuronal activity. Increases and decreases secretion of insulin. Elevated blood food, elevated blood glucose, elevated blood amino acids, elevated blood fatty acids. That's pretty much what you need to know, really. Why athletes use insulin and growth hormone together? Because separately they simply don't work. Okay? Because insulin is needed to transport amino acids into the cell, and growth hormone will stimulate protein synthesis from those amino acids. Does that make sense? That's what we just talked about. So growth hormone stimulates protein synthesis from amino acids. Amino acids get into the cell because of insulin. Does that make sense? Diabetes, no two types. Type 1 is juvenile, genetic, lack of insulin. Type 2 is lifestyle, later in life, insulin is high, but people develop resistance. Okay, I want you to know the idea of insulin resistance. Okay, so in a person with type 2 diabetes, what do you expect in the blood? Like, what levels of glucose are up? Levels of insulin are up. That makes sense? That's fancy word. Comorbidities. What are they? Pretty much, people with type 2 diabetes, what do they suffer from also? Often, obesity, hypertension, hyperlipidemia. So it's a lifestyle choice. Treatment options for type 1. Insulin. For type 2. Lifestyle change, yeah. Diet, exercise. Insulin too, but diet and exercise work much better. Glucagon chemically is the protein. What type of signaling does it trigger in the cells? Second messenger, okay. What stimulus? Low blood glucose. Metabolic changes. What we just recited, okay. Glycogenolysis, gluconeogenesis, lipolysis, all of it is stimulated by glucagon. Am I clear? Understand that? Okay, 4 gonadal hormones. focus on males in terms of the regulation. Hypothalamus produces gonadotropin releasing. What does pituitary produce? LH and follicle stimulating hormone. In males, follicle stimulating hormone stimulates spermatogenesis. In females, the name, people, oogenesis, follicle maturation. Luteinizing hormone in males stimulates production of testosterone. In females, production of estrogen. Okay, that's basically the axis. In males, what are the effects of testosterone and inhibin on the first two hormones of the axis? Testosterone, what does it do to? Yep. It inhibits FSH and LH and inhibits gonadotropin releasing hormone and stimulates gonadotropin inhibiting hormone. What is the function of testosterone in males? What do you need testosterone for in males? Like, imagine there's a male that doesn't produce luteinizing hormone. Will this person have functional sperm? Nope, because testosterone acts on seminiferous tubules. Testosterone is necessary for the sperm maturation. Okay. Okay. Well, sperm cells or follicles, testosterone, estrogen. Now, general effects of testosterone. Secondary sex characteristics, deepening of the voice, growth of muscles and bones. Oily skin, you know. Recall your puberty. If you're male, or recall the puberty of males that you used to know, or know, you know, if you're female. Does that make sense? Female gonadal hormones, female body, widening of the hips, growth of the breasts, growth of the reproductive organs, enlargement of uterus, skeletal system, calcium retention, bones become stronger, cholesterol is healthier, but there is estrogen presented. Just know. Let's leave it to like very last moment of this semester. Okay? The female HPG axis, let's just leave it for the reproductive. Seriously. Too much on your plate right now. Okay? For regulation, stick to the male one. I promise there will be no questions on the regulation of female HPG axis. Okay? Pineal gland, melatonin, precursors are tryptophan and serotonin. Function is circadian rhythms. Light inhibits production of melatonin. Thymus controls the maturation of lymphocytes. Okay? Thymus degrades with a lifetime. And P, blood pressure regulation, right? High blood pressure, more atrial natriuretic peptide. People start to excrete sodium in urine. Sodium attracts water. Water leaves the body. Blood volume decreases. Blood pressure decreases. Does that make sense? What you need to know for these three: histamine and gastrin increase stomach activity, stomach digestive activity. Ghrelin is the hormone of hunger, produced when you're hungry. Stimulates appetite. The main stimulus for these two guys, food. These four, food in the intestine stimulates production of these four hormones. Affects these two, all you need to know, increase the activity of the intestine and the pancreas, digestive activities. GIP and GLP increase insulin production. Renin is produced by kidneys in response to low blood pressure. Renin activates angiotensin, which is produced by liver, and the long-term outcome is elevated blood pressure. Does that make sense? Erythropoietin is produced by kidneys. What can stimulate erythropoietin? Hypoxia or low it's hypoxia, but low red blood cells, which may result from anemia or blood loss. Okay? Does that make sense? Bones produce this, guys. Excretion of phosphate. That's all you need to know. Osteocalcine. Insulinotropic hormone. Increases insulin production. Somatomedin. IGF. Another name for insulin-like growth factors. Liver. Growth hormone is the main stimulus. For effects of insulin-like growth factors, refer to our discussion of growth hormone. What stimulates conversion of angiotensinogen to angiotensin 1? Renin converts angiotensinogen into the angiotensin 1. Downstream effects of angiotensin 1, elevated blood pressure. You see, those are like repetitive topics that we talk about. Thrombopoietin is produced by liver. What stimulates its production? Low levels of platelets. The effect? Normal levels of platelets, elevated. Hepcidin, it's a typo here. Hepcidin is produced by liver and controls, all you need to know, controls iron metabolism. Okay? Now for leptin. Stimulus for leptin is food that you eat, and it results in the feeling of satiety, so you stop eating. So if somebody doesn't have leptin produced, what this person is going to have? Somebody doesn't have leptin. Obesity, because there's no control, right? Now, obese people, do they have elevated leptin levels or low leptin levels? Elevated. Why? What do they develop? Because they constantly overeat. They develop resistance to leptin. Does that make sense? So remember that feature. Ghrelin is opposite to leptin. Adiponectin is produced by adipose tissue and liver and muscle, actually. Main functions: anti-inflammatory, cardiovascular system, increased cardiovascular health, decreased inflammation, decreased lower risk for diabetes development. Okay, improves glucose metabolism. In obesity, leptin levels are elevated and adiponectin levels are decreased. Questions? I think it was pretty comprehensive, wasn't it? Break.